Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we jump into this new episode, we have exciting news to share with you. The new Dr. GPCR ecosystem platform is almost ready for launch. What is it about, you ask? Well, think about a place where GPCR scientists, trainees, and GPCR organizations can thrive and where it's all about science and GPCRs. Access is restricted to members of the field, and each sign-up will be approved accordingly. Once your sign-up is approved, you'll be able to enter the ecosystem and interact with GPCR colleagues like never before. You can still sign up to be an ecosystem beta tester. We are going to open the ecosystem slowly to ensure that you get the best experience. Please keep in mind that the Dr. GPCR ecosystem is a unique place, a first and one of a kind, where we want to make sure that you get the best experience out of this new world. Visit drgpcr.com/ecosystem to start your GPCR journey. Also, make sure that you mark your calendar for the third edition of the Dr. GPCR Summit. This year, the summit will be held between October 10th and 16th. Stay tuned as we are working on the program for the summit. Visit drgpcr.com to find out about all our activities. And now, let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR. I am excited today to have with me Dr. Aylin Hanyaloglu. She is a faculty member at the Imperial College London. Aylin, so great to have you with us today.
Thank you so much, Yamina, for having me.
I'm so excited to talk to you, especially that, we've been trying to set this interview up for so many times and things happened. So when we nail down a date, it just, extra happiness to be able to do this. So tell us a little bit about yourself.
So I'm born in London. My kind of sort of science journey has, even though I'm still in London, it's kind of been a circuitous route, but also lots of twists and turns in that road, I guess, geographically and kind of career-wise and emotionally in terms of how I've got to where I am today. So I guess I wanted to be a scientist, kind of didn't really kind of come into my radar probably until I got to undergrad. I actually wanted to go into medicine, so I've always kind of had a love of biology in high school and had some great teachers, and really kind of like fascinated about how our bodies worked and kind of trying to understand it from that perspective. And so I originally wanted to apply for medicine, and I did, and I got an offer for medical school, and I think with the pressures of all of that from having that, and I know my family were kind of like really excited about that, and then it got to the exam, and I just had then those moments where it's just completely, my mind went blank. So I did not do well in my exams. I didn't get the grades that I needed to get to medical school. So I had a decision of, I could have gone back and retook those exams, or go and take another path, which was to apply for an undergraduate program, and with the possibility of then moving to medicine at a later point from that. So for me, I couldn't bear the thought of going, staying another year in high school to retake those exams. So it felt like I wanted to move forward. For me, moving forward was as to go to an undergraduate course. So I went to King's College London, and I did a bachelor's degree in human biology. And it was in my second year that kind of things changed, where I kind of dropped that idea of wanting to do medicine. And I put it down to a particular course. So in the second year, we did reproductive biology, and it was led by an amazing teacher and scientist, Lynn Fraser, who sadly is no longer with us. And she was really inspiring. And at the time, I had a family member who was going through a lot of rounds of IVF, and she was really struggling to have her own children. So with that combined, I really wanted to know more. So I signed up to do a summer studentship with her in Lynn's lab. And I got the research bug from there. So even from that short time I was there in the summer, I actually incorporated some of my data in a paper and get like a publication out of from that really short period. I was like, wow. And so I did my undergraduate project with her. And from that point, it's like, yeah, okay, I want to do a PhD. I want to do more of this. So the project that I was doing was working with spermatozoa and this TRH-related peptide. So unbeknownst to me, the kind of GPCRs were coming to the picture, but I wasn't really aware of it at the time. And so I applied for a PhD in Edinburgh. So at the time, there was the Human Reproductive Sciences unit. It was the place to go in the UK if you wanted to study reproductive biology, and it was very internationally renowned. And I wanted to move more into kind of the female side of reproduction, so I applied for a project that was looking at angiogenesis within the ovary. And when I got the call to kind of get the outcome, I was told it was a real sort of tie between me and another candidate, and they decided to go with the other candidate in the end. So I was really disappointed not getting that as well. But as a result of that, from the interview that I had, someone on that panel spoke to somebody else who was also looking for a PhD candidate in the same institute, and from that, I got a call saying, could you come back up again and we'll have a chat? And this was Karen Edney, and she was working with pituitary G-protein coupled receptors. So that was my first introduction to the GPCR field, working with GNRH and TRH receptors. And then from there, during the end of my first year of my PhD, she moved to Australia. She said it was just a sabbatical, guys. But then she called me up about a few months in saying the lab that she's in, they're doing yeast-2 hybrid screenings, which you get to come over and you can do yeast-2 hybrid, learn about this technique. It was at the time when everybody was doing yeast-2 hybrid screens. I was like, trip to Australia? Absolutely. I'm not. I was going to go for like 2 or 3 months, learn his technique. It would be great. And then when I arrived in Perth in Western Australia is when it kind of became apparent that her move was a bit more long-term. And this was kind of my time to decide whether I wanted to continue my studies in Australia or go back to Edinburgh. So I ended up deciding to stay in Australia, and actually ended up graduating from the University of Western Australia from there. And then I started to apply for postdoc positions, and I knew I wanted to go to the States, and there were particular groups that I'd fallen in love with the work they were doing. So for my PhD, I was working on GNRH and TRH receptors. We were starting to look at GNRH receptors, one of those GPCRs that doesn't have an intracellular CTEL. So it was one of those first receptors that we reported that doesn't recruit arrestin at the time in the sort of late 90s, early noughties, when everyone was starting to look at arrestin recruitment and which GPCRs use arrestin. So this was one of the first ones that showed that didn't recruit arrestin, and it was through this lack of the tail. So we were looking at the sort of the trafficking and the how internalization was regulated for this receptor, but we were also interested to start looking at dimerization at that time. And so the kind of two groups from that I was interested in was Michel Bouvier and also Mark von Zastrow through that work. So it was a really tough decision. So I interviewed at both places, and yes, it was really, really difficult. It probably didn't help that I visited Montreal in January.
Yes,
and then went to California sunshine. But yeah, so I chose to go with and work with Mark. Not for whether Michelle is amazing, such a gentleman, and it was such exciting work that they were doing in both areas. So I thought I was only going to be in San Francisco for a couple of years. It ended up being 5 years there for a postdoc. And then it was kind of deciding, those last couple of years, deciding what was I going to do next? Is it something that I really do? Did I want to continue down the academic path, start my research group? Did I, considering industry as well? And then I was even thinking about, potentially, is it ready? Am I ready yet for independence? Should I be applying for another postdoc? And I have to credit Mark for pushing me in that direction, saying, yes, you are ready. If there are opportunities, like go for it. And so there was a position that came up at Imperial in the Institute of Reproductive and Developmental Biology. So after kind of stepping away from the repro field, I kind of felt like I wanted to kind of go back into there. That was kind of my first love. And so that opportunity came up for a position there, and that's what I took, and I was fortunate to get there. And I've been in Imperial ever since that time.
I love it how you really put it in there that you visited Montreal in January. And I feel after 16 years in Montreal, I'll never forget, we were going to a conference. Actually, it was a Gordon Research Conference of Molecular Pharmacology in LA, in Ventura, and it was in January, and it was minus 40 Celsius in Montreal, and we were all bundled up, and the flight was delayed because the plane, whatever parts were frozen, so we were delayed 6 hours, and then we flew to LA and we got out, and you could see everybody in our group who went to the conference, getting their coats off, putting on their sunglasses, and being like, finally. So I feel
yes, coming from Perth, living in Perth for 3 years, West Australia, with the sunshine and the heat. Yeah, so it was quite the shock.
But you made, I want to take us back to the beginning of your journey, when you mentioned, not being stressed out and not getting into medical school, but not performing well on those exams. Looking back, do you feel lucky that this actually happened?
Absolutely, yes. You kind of feel now that was the kind of the right path to take. I'm not sure. I mean, it's kind of one of those sliding doors moments, right? You don't know. What would have happened? If I had got into medical school, would I have actually continued it? Maybe I would have fell into doing, science. I don't know. But for me, I'm very happy with the journey. It's, those particular times when you're in that moment, you're absolutely disappointed, really upset. You think your kind of whole world has kind of ended. But you kind of, now looking back, it's, always one opportunity closes, then another one opens, right? So it's
The reason I was bringing this up is thinking about me, but I'm pretty sure that I'm not the only one who was like this as a graduate student. As when something didn't work back in the day, it was the end of the world, as you mentioned, in that moment. But that's also a life-determining or life-changing opportunity as well. So if something doesn't work, yes, it's okay to feel sad and disappointed, but it's not the end of the world on that end.
Absolutely. And I'm also kind of glad to see that now the kind of emphasis on exams is kind of being reduced in terms of how we learn as individuals. So the emphasis more about transferable skills, and that skill-based learning encourages that deeper learning. So it's not all about how you perform at that particular day, at that particular time. It's kind of how you're growing as your skill set and as a person, and how these are lifelong skills that you develop. So a lot about like how undergraduate, postgraduate courses have more of an emphasis on that now.
I think that's a necessity. And the second kink in your road when you didn't get that position for the PhD, but then you still did your best, and then out of that performance that didn't go your way, you got a call back for another lab.
Yes, exactly.
Which I think it's just so fun to hear about, because these things happen, but I don't think we talk about them often enough.
No, I think when you talk about it, it sounds like been a very direct linear pathway, and it's just all kind of just worked out. But I think most people's
Beautiful
stories are not like that at all.
I think another highlighting moment where it must have been disappointing at that time because you put in effort to go there and interview, and it didn't work out and they didn't pick you, but then it kind of defined your path.
Yes, absolutely. Yeah, and how I got into GPCRs. So that was that turning point.
And it also took you to Australia. I mean, I think it's a good thing.
Oh yes, absolutely. Amazing experience, amazing place. It was quite the learning curve. So I kind of was, when I was joined the group, it was a sort of a large established group. It was it was a centre, so it was kind of core funded from the Medical Research Council. And then to have my supervisor make that decision, and part of it because of her partner as well, had got this amazing job opportunity in Australia, to restart everything from scratch. So it was literally just me and her, and we had to kind of use somebody else's bench initially until like her space was sorted out. But then I got involved in actually starting up a lab, because it was just me, and then we got students, and then I had to train. So I was kind of like, I guess, the baby initially with this established group, and then I had to kind of help her to set this up. Yeah. So that was another great learning experience as well. It was very, very interesting being in Australia. You really feel the isolation sometimes as well. You had to really prepare in terms of anything that you wanted to order in, because it could take just weeks to get to you. It kind of feels like that now, actually, at the moment with COVID and Brexit. Just you have to plan now to get anything in. But yeah, so it was that aspect. You felt that isolation, but felt really lucky of where you were. I mean, being able to just go to the beach for a swim and then come to the lab in the morning. I mean, it's an amazing, special place.
It has its own charm, and it kind of reminds me of a little bit of the kink in my road. So I also applied to be a summer student in Michel's lab. And at the time, he was head of the biochemistry department, and he must have liked me, but there was one spot and we were at least two. And interestingly, I learned that I wasn't picked because and I remember this like it was yesterday. I was at the library at the University of Montreal, and one of my friends and colleagues, who is now actually a professor at the University of Sherbrooke, Martin Audet, jumped up his chair saying, "Michel picked me. I'm going to spend the summer working in Michel's lab," without him knowing that I had applied as well. So I was like, okay, I guess it's not me. But then again, there was a new professor coming in at the department, and Michel introduced me. The same day I got the email, he said, "I really liked you, but I had to make a tough choice. Then again, I think it would be a good fit for you to talk to this new researcher coming in." And just similarly to you, I got involved into putting together a lab, mm-hmm. which I think was a priceless experience. When I went to interview for my summer internship, which turned out to be a master's and a PhD degree in the same lab, that was the PI, the phone. I don't think he had a computer, or maybe he had a computer but no phone, and bills everywhere because he was ordering up stuff.
Wow. Yeah.
So the take-home message for whoever's listening is it's okay if things don't work out the way you envision them. The goal is not to give up.
Yes, absolutely. Couldn't agree more.
Love it. I love it. So you mentioned that you worked on different GPCRs. You're well traveled, definitely. Let me ask you this, and I ask this from everybody. What is your favorite GPCR to work on? Or family of GPCRs?
I would probably have to say the gonadotropin hormone receptors, which these are GPCRs that we are currently working on. So I have been working for nearly 15 years now, and the reason that I picked them, I wouldn't say that they're not the easiest receptors to work in, especially after doing some work with beta-2 adrenergic receptor, where you feel like everything works, most well-behaved GPCR ever. Like every single assay you do, it works perfectly. So there have been, like, technically have been challenging receptors to work with, but with that, they've also opened up these kind of new mechanisms and pathways that we weren't expecting to uncover by studying these receptors. So they've taught us a lot of lessons through studying these two G protein coupled receptors, and a lot of it has kind of been very serendipitous in terms of we were looking for other things and we kind of stumbled across these other aspects. We were like, okay, well, I'm not really sure what this means. We'll have a look a little bit further, and then it's kind of opened up a whole other avenue of research. So I guess sorry.
Good. No,
it's
good. As I was just going to say that, so basically you started working on the GNRH in Australia and then took a break in Mark's lab, or did you continue working on this in Mark von Zastrow's lab and then kind of have you traveled with this receptor or your work?
Yeah, so I was working with pituitary, GnRH, and TRH receptors in my PhD, and that was really, and we had some other side projects with calcium sensing receptors, but it was with those two GPCRs that I stayed with just for my PhD, and so we were looking at how using these two receptors to kind of understand how arrestin may or may be recruited to some GPCRs and not other GPCRs. But we were also looking at receptor dimerization. That's kind of where we first started using BRET in a plate reader format. So it was currently kind of came up with the idea of kind of what we currently have as the Mithras, the Berthold Mithras plate reader. So it was after Michelle published that very seminal study in PNAS in 2000 with showing using BRET to look at beta-2 adrenergic receptor dimerization and ligand-induced association with arrestin. That was done in a top count machine. So if you've done any kind of like scintillation counter, I don't know if you used one of those.
I did, but it was a long time ago.
Long time ago.
I think I got into the field when that was, phasing out. And we were going into the plate, the Mithras and using BRET-based, live cell assays.
Yeah.
And it was really at that junction when I came into the field.
Yeah. So my PhD supervisor wanted to adapt the kind of plate reader luminometer. So she contacted Berthold to see if they could put filters in to their machine, which they agreed to. So I have to say that first kind of prototype, literally, this would only be relevant to kind of any UK listeners. It's like a Blue Peter project, because it was literally like cardboard and sticky tape of how this kind of mechanism for switching the filters when you looked into it. It was very crude, but it worked really, really well.
And you can hear it if you pay attention when you're reading your plate in the Mithras.
Yeah,
you can hear it, but you can also set it on how many seconds you'd want that to read an age for, and then you can hear it. And sometimes it did happen to me in the lab when towards the end of the plate it was going faster, and that was always concerning. But you could hear the filter switch happen. I think you can still hear it.
Yeah, so that's why I kind of had that interest between those two labs. But I wasn't necessarily tied to kind of staying with those particular GPCRs, so I didn't get a chance to kind of continue working with those particular GPCRs when I moved to Mark's lab. With Mark's lab, there was a lot of work with the beta-2 adrenergic receptor, but he has also a strong interest within the opioid receptors as well. But it was all primarily working on mechanisms of membrane trafficking. So I had a sort of a break from the kind of reproductive relevant GPCRs or endocrine GPCRs to work with the beta-2 adrenergic receptor, and then coming back to starting my own group and coming back to a reproductive biology environment, going back into those GPCRs was like a natural step. But these were with working further down the kind of hypothalamic pituitary gonadal axis with the glycoprotein hormone receptors, so the gonadotropin hormone receptors, LH and FSH receptors. And that was a time when I was, we had a group who was working alongside me. So when I started, it was just literally just me starting like the lab. I was kind of taken under the wings of Professor Ilpo Hutanimi, who was doing a lot with the glycoprotein hormone receptors at that time. He was an endocrinologist, and he was starting to go into the kind of molecular side, but not so much. So I was working with him, and that's kind of how my work with these GPCRs kind of came about. So combining the work that I'd done with the beta-2 adrenergic receptor and trying to apply it to understand these GPCRs and how that might change their function in vivo in terms of reproduction.
I love it. So for those of us who are not well versed in the LH and FSH receptors and GPCR receptors, if you had to compare, obviously the beta adrenergic, as you mentioned, I think it's like this most beautiful receptor because it does everything so well and so easily. What should we know about the LH and FSH receptors?
So LH, so luteinizing hormone and follicle stimulating hormone receptors. So they are key parts of the hypothalamic pituitary gonadal axis. So they're absolutely essential for reproduction. They are the targets for anyone going through any kind of assisted reproductive technology. So this is kind of how you would try to stimulate the ovaries. You could go through a protocol of being able to increase the number of follicles and then being able to ovulate. And so that process, in terms of any kind of fertility program, they are the targets of. Still using recombinant hormones rather than anything like a small molecule based approach. They have been implicated in other tissues outside of the reproductive axis. So there's been a lot of work from other groups with looking at FSH receptor in bone and adipose, and the story around FSH being like an aging hormone. So there is work that it might actually increase osteoclast activity and in terms of like fat storage. So these kind of changes that happen during menopause, where you have changes in the levels of these hormones, you have high FSH, increasing levels of FSH, and these might act in other tissues. There's a whole other story that's emerging potentially from the cancer side, which I would say is quite still controversial. Actually, anything extragonadal of where we don't normally expect to find these receptors, there's always still a discussion around, is it real or not? And that partly comes down to some of the tools perhaps that have been used historically to look at this, but I feel that that's probably going to change. So these receptors are they have obviously key functions in terms of our sort of reproduction. That's important as a species, but for me, in terms of from the molecular side and actually trying to understand it from how kind of cells respond to their environment, they've been for me quite a game changer in understanding new pathways. So with the LH receptor, it's kind of uncovered a whole endosomal trafficking system. So there's been obviously an interest in the past decade of how where a receptor is changes the activity of that receptor, and that GPCRs don't have to just signal from the plasma membrane, they can signal from other parts of the cell. For the LH receptor, when we were trying to look at its trafficking, the kind of first kind of initial experiments we were looking at to try and do some of the work we were doing, the beta-2 adrenergic receptor, my initial thoughts when looking at this receptor doesn't really internalize. Like you couldn't really see much. It wasn't robust internalization. But actually what we've kind of then uncovered is actually it's very good at doing that. It's actually very good at recycling is also what it's good at. But it actually traffics to a very different compartment to the beta-2 adrenergic receptor in terms of the kind of endosome. So we've kind of found this new compartment we call the very early endosome, and how certain GPCRs must have to traffic there for its signaling in order to have its normal activity and function. So that was one of those kind of serendipitous findings. We were looking for something, a receptor based on the work we've done with the beta-2 adrenergic receptor to look at those pathways, but it was behaving in a very, very different way.
I love it. I'll let you take a sip of your water
Okay.
while I really love it because this is it kind of confirms. I when I talk to people and they ask me about GPCRs and receptors, and I say, well, they're all beautiful, they're all different, they all have their quote unquote personalities. And once you start working on the GPCR, in theory, it seems simple to understand what signaling pathways it's activating, but then in practice, you kind of have to get to know your GPCR because no two GPCRs are similar. Sometimes you get lucky and you work with a receptor like the beta adrenergic and everything is beautiful. And then sometimes you find a GPCR where, in theory, it should be easy peasy, but then you have all of these difficulties and the signal is not what you've expected. And then you have to dig deeper, like with the LH receptor that you mentioned, and figure out, okay, so what does happen in reality and why it's different, or
Yes.
how is it different from your
Yes.
quote unquote regular prototype GPCR?
Yes.
So here's my next question with that. What kind of methods do you use typically in the lab to uncover the function of the LH receptor, for example, where you were able to identify that it's very rapidly recycled, most likely so rapidly that you don't see the difference in receptor expression at the cell surface. But what's the methodologies what's the tools that you've been using to uncover these mechanisms?
So a lot of this is all image based, and I love microscopy. I think that's kind of come out from actually since my undergraduate project, which was all kind of microscopy based as well. So that's kind of continued on through PhD, and especially with Mark's lab as well. And if you're wanting to look at receptor trafficking, sort of assays where we can see the receptors in any way we can see the receptors and being able to quantitate that. Is the kind of the primary methods that we are using. So that would be both confocal and turf imaging, both live and fixed. But we also use, for say, rapid recycling, we use the receptors that are tagged with a pH sensitive GFP, the ecliptic GFPs. So these are internally tagged, so they are fluorescent at neutral pH, and then they quench in the kind of more acidified environment of the endosome. So that allows you to measure kind of the real-time reinsertion of the receptor back into the plasma membrane. So you can see these almost like, they call them puffs. They're like little fireworks in the membrane as they explode. They're quite fun movies to watch. But you can get very rapid kinetics of receptor trafficking as one readout. As with this compartment that we identified is much smaller than your classical early endosomes. It's about a third of the size smaller. So we're sort of started to move into super resolution territory to get the resolution that we need to kind of start looking at. If we want to start looking at single endosome dynamics, potentially even microdomains on those endosomes. So we've done a little bit of STED microscopy to look at some of this, but unrelated to membrane trafficking, we're also using super resolution imaging for receptor complexes at the plasma membrane. So looking at monomers, dimers, oligomers, etc., and that whole landscape, and how receptors are organized at the plasma membrane.
I love it. I have zero or not so much experience in microscopy, and it's something that I've always wanted to kind of conquer. But then when we got the Mithras and the Brett sensors, which shifted to, Brett here and there, mm-hmm. I think it's just it must be such a satisfying feeling to actually see an image come out of an experiment and work through that image, obviously with the correct controls and making sure that everything is working correctly, but still getting an image, seeing it, or yes, even watching a movie of as it is happening. I think it must be very satisfying.
Yes, I've always really enjoyed it. Even obviously we do assays where we have we do Brett-based assays, but also kind of more kind of classical pharmacology assays, dose response, etc. You have a lot of numbers, which is just fine. But I think sometimes to have that other additional like validation where you can actually see something has happened as well as the numbers telling you as well. I I like also being able to kind of to see things as well. Yeah.
Love the double validation aspect of it, definitely, because numbers are great, but you're just imagining you're just experiencing, quantifying the consequences of the event happening in your cells.
Mm-hmm.
But seeing it on a video, I think it's just it's the next level of satisfaction there scientifically.
Absolutely. And then the sort of biosensor tools that obviously have come out in the past few years and are probably likely to continue are going to help with that from an image-based perspective, especially with like the nanobodies, for example, and looking at different activated states of both receptors and G proteins, and where those things happen in a cell. It's it'll be more work to do, more fun stuff to do, definitely.
That's awesome. From a disease perspective, you mentioned that there is this controversy, as to what disease, in cancer, and then in aging, you had mentioned as well for the LH receptor. Yeah. What do we know for sure for the LH and FSH receptors in a disease context?
So there are mutations that have been identified in these receptors, and I have to say that they are quite rare to have like activating and inactivating mutations. So inactivating mutations obviously cause infertility, and there are often cases of activating mutations that cause precocious puberty. And like I said, these are quite rare conditions. FSH receptor is one of those that has a number of single nucleotide polymorphisms. I think they probably haven't been fully explored. There've been a lot of association studies that might dictate, depending on groups, there have been links to how someone might respond in an IVF procedure, how well they would do, to links with preterm birth. There's been a lot of associations, not kind of direct kind of studies that have been done. We do know that these receptors are involved in polycystic ovarian syndrome. So this is quite a common endocrine disorder in women. So in the UK, about 1 in 10 women will have a PCOS, and it's quite a complex syndrome. But there's really been studies showing that the kind of activity of, say, the LH receptor is like almost like hyperactive receptor. And we've got a current project with Steve Franks. We're trying to look at apply some of these kind of mechanisms of what's with the trafficking of the receptor and how that might be altered in these women. But there are kind of known diseases where these receptors are involved. But I have to say, as a group of GPCRs, we're quite poor on the kind of drug small molecule side compared to other receptors. So I'm quite envious compared to other GPCRs that have quite the menu of agonists and antagonists. That's starting to change, but there's certainly nothing that's been used clinically beyond the native, like the recombinant hormone. But there's kind of been, people have been trying to create selective allosteric molecules, these receptors, trying to make them orally available. So instead of women having to self-inject these hormones, that they'll be able to just pop a pill. But those I think they started happening, and then kind of I think that whatever reason, sort of interest within the company themselves, things change in terms of where that company's going, and things haven't progressed. But I hear in the grapevine that might be changing with certain companies might be jumping on that again. Mm-hmm. Certainly as a cell biologist, having these pharmacological tools is great to be able to try and unpick some of the mechanisms that we're looking at. And we started to work with some allosteric small molecules. They've been mainly focused on FSH receptor, but there is a few for the LH receptor. But yeah, that's definitely one of the areas I think that could be improved is having those pharmacological tools.
Well, if anyone's listening and feels like collaborating and developing these pharmacological tools, they'll be able to find you. But I think that this is so interesting. I have many questions. I'm trying to organize them in a logical order. So you mentioned some of these mutations, and some of these are inactivating and activating mutations in the FSH or LH, I can't remember, but then, and this is very rare. Overall, if you're looking at modulating these receptors is using the native hormone that people get to inject, for example, for an IVF protocol. But in these, what from a clinical perspective, would we want to inhibit, or would we want to activate, or we want to modulate these receptors? What could be the most urgent or the most useful mechanism?
I would go for the modulate, because you would want to be able to, unless you're not downregulate, because you'd have to consider that there would be obviously your own LH and FSH in your system. So you'd want to modulate. So the example, say for PCOS, women with PCOS, where it's kind of an example of an overactive system. So you would have, and these women, a lot of them have increased levels of LH that they're pumping out from their pituitary, and being able to modulate that, so as like a potentially as a negative allosteric modulator. But then I would go one step further, because we kind of are moving as a field away from considering these receptors as kind of the on-off switches, and we want to go more pathway selective. So a lot of these, these GPCRs, as for other GPCRs, they are pleiotropic. We know that where they signal from matters. So if we kind of have to be more on the modulator side to get the specific effect clinically that we want in terms of specificity and the efficacy.
Yeah. So we would want to be surgical, basically.
Yes. I know that from a drug company side that they don't want to think about it that way.
Well, yeah, it's hard because
that's too hard, but
yeah, I think it's hard because until you don't know where to hit, and you just hope that you're just going to do something that modulates. But then again, a lot of questions come into mind in these patients. Is there a trend? Is it always this amount of extra LH, or is there a range? Because then you're talking about personalized dosage or personalized medicine there. So I think there is a lot of questions that remain to be answered. But from a perspective of basic research and getting tool molecules, do you know what has been done? And if you had a magic wand, what kind of molecules would you like to have to use as a tool molecule?
Yes, so the kind of development of the kind of small molecules has kind of been sort of 2 decades in the pipeline. I would say it started a company called Organon that kind of merged into another company and then merged in, I think it kind of finally with Merck currently. But there has been developments of agonists. There's been, I think harder has been antagonists. Specificity has been an issue in terms of being able to discriminate between LH, FSH, and its related glycoprotein hormone receptor, which is the thyrotropin stimulating hormone receptor. So those 3 GPCRs kind of sit in their same little subfamily within the class A group of receptors. So they all have this large extracellular domain. So they look more like a class B GPCR rather than class A, but they are, yes, they've got their own little subgroup within the class A family. So having molecules that have been able to discriminate between those 3 has, I think, been of a challenge. And it is something more recently. We've worked with a company that unfortunately no longer is around. It's called Tocopherix. We had these FSH receptor small molecules, and we had some different chemical classes, and we've been doing some work around trying to understand how they might activate the receptor in different ways in terms of both bias and spatial control of signaling. So there is people who are starting to make those tools. The kind of missing element is to know what exactly do we need, because we're uncovering more about how these receptors function, and we need to get into those relevant cell types, the physiologically relevant cell types, and to try and understand what is happening there. And then we can tailor it to say, okay, this is the kind of molecule that we want with this specific activity. And as you say, it might have to be tailored. There might be, say, women going through infertility treatment who may be like poor responders to FSH. So those are known who they respond to as they get given FSH to kind of stimulate their ovaries. They don't respond so well in terms of the number of follicles produced. So there may be a kind of case for that, maybe certain individuals who have a more kind of tailored approach to their regime.
Yeah. Yeah. It's, I think it's fascinating. I always, in undergrad, I had this, the name of the class was Molecular Physiology. And I really loved the series of courses on hormones and all of these pathways and the reproductive system and how everything is precise and timed in the best case scenario when it comes to hormones. And, there are these cascades. And I feel like it's like this relay sprint where, you pass the baton from hormone to hormone in order for them to do what they need to do. And I think it's just fantastic. And I feel like hormones, I find them dangerous in the sense that they have to be on time and precise. Otherwise, it just messes up the whole system. It has to be well organized and strategic about when and where these hormones act. We've talked a little bit about the tools that you feel like would help study and better characterize these receptors, but it's kind of the egg and the chicken, because you need to figure out what is going on in order to then say, well, surgically, this is the outcome that we want, and then get the molecules. But without the molecule, it's kind of difficult to figure out those mechanisms of action.
Yes. Yeah.
In a broader term, and again, I asked this from everyone, and I know the answer to this question is yes, but I would love to hear more beyond the yes. Do you think GPCRs are still good drug targets?
Well, obviously, it's going to be yes. I don't think you've ever had a no, right?
I don't think so. No. Well, no, because I only talk to people who actually are in the field. Yeah. So if they're in the field, then the answer is yes.
Yes, I definitely believe they are a good target. And what actually is always amazing to see is what kind of new targets that people are coming up with for these receptors. So we know that people are extending beyond the orthosteric site for these GPCRs. But that kind of term for allosteric is not just on the kind of extracellular region, and people targeting the intracellular sides. We are kind of thinking about it not just from a single GPCR, but how that GPCR is kind of in its environment in terms of other proteins around, and kind of thinking of it as a complex, and that's a way of tailoring the specificity for the drug response that you want. So there is work in terms of looking at heteromer targets, for example, and not just for another GPCR. I mean, there's work obviously you work with in terms of crosstalk with receptor tyrosine kinases as well. Yeah. But I think they always will, I think, remain a good drug target. They have the track record for it. There's still a lot of GPCRs that haven't been targeted, and so I think that it's a big yes.
And it's funny because when I put together these questions, I hadn't thought deeply enough about, how does the biotech and pharma world function? And sometimes it's difficult to figure out if a company is working on the GPCR, because whatever is available online is about disease area. Yeah. And the beauty of GPCRs is that they do everything. So at any time, anywhere in the world, any company might think, oh, we are focusing, I don't know, on immuno-oncology. What GPCRs? What are the GPCRs that we could be working on? And there you go, work on GPCRs.
Absolutely. There's always a GPCR for that. Whatever you're working on, there's a GPCR.
I know. I feel like whatever you do, there is a GPCR involved. And when I talk to people who are not in the field, and I introduce myself and I talk about GPCRs, the tagline, and maybe I'm going to share this with the world, maybe others want to use it as well, but I always tell people that you can see, smell, and taste thanks to GPCRs. And I think that's like the most tangible thing, but they do so much more We know a lot, but we don't know nearly enough.
Oh, absolutely. Yes, it's going to keep all of us very busy.
I think so. There's more kinks in the other people's trajectories coming for sure, thanks to GPCRs. So we've talked in the beginning about how you got into the field and how your trajectory led you to where you are today. If you had advice to give to junior scientists, to trainees who want to either contribute to the field or just want to be quote unquote just scientists, what would that advice be?
I think my advice would be is to find your champions. And there might be multiple of those, and I feel lucky to have had a very strong female role model, like a scientist that have inspired me during my training. But actually, on reflection, in terms of my champions, they've been male and female, and they've come from different areas and different parts of my life. But they've all been. Everyone needs that support network. So they may not necessarily have to be your advisor or your PhD supervisor. There are other people around you that are there who are with their banner saying go go go. Yes. So I think it's finding those champions. There's been kind of certain key people I think that have influenced me and have really kind of helped me to progress to that next level. And when you kind of wanted to kind of throw in the towel and everything, you need that support network. It's tough. I mean, science is hard to do. These are really difficult things, and so I really value that support network. We talk about it takes a village to raise a child, and I would say it's as a trainee scientist, it's the same thing as well.
Agreed. I love it. And it's funny because I was browsing social media, I think it was yesterday, and someone shared an interview with Bob Lefkowitz from 2012 when he won the Nobel Prize. And I think he said something around, if they would drop him on a deserted island with all the lab equipment and all the time, he would be really sad because to him, science is a social activity. And I couldn't agree more. It's not one person pipetting alone in the lab. It's really a village. It's these conversations that help advance the field, that help form relationships. And I think we're stronger together.
Absolutely. And we are, and it's on an international level too, as a community, which I think is such a big plus for what we do. I always kind of thought, you have say to other people, you have a PhD as a scientist, you get an international passport to work anywhere you want. And perhaps these days, I know it's harder, but we've probably through these kinds of interactions through Zoom and Teams, we've continued, and perhaps some of these international collaborations have grown. We've through this format, we've written grants and put together conferences through people in all four corners of the world. And so it's definitely on a big scale, this kind of this community. So it's that for me, that's a real plus for what we do, that we get to work with so many people from so many different backgrounds and different parts of the world.
Yeah, and we all have GPCRs in common, and we all think that they're good drug targets. Fantastic. We talked about, as I mentioned previously, about the rejections that happened in your career and how that shaped you as a scientist and as a person. Are there any top aha moments from your career that you think you can talk to us about, whether it's scientific or on a personal level, that actually shaped who you are today?
So I think I mentioned perhaps already, one of these kind of aha moments is kind of starting early on from my group at Imperial and working with the LH receptor. And it was just as simple of kind of an experiment where it was like, oh my goodness, what is this? And it was just that kind of image-based study just to show the LH receptor is in these little kind of teeny tiny endosomes, and that was very different to how the beta-2 adrenergic receptor was behaving, and that kind of was something that was the start of then a whole other program of work from that point. And in terms of other aha moments, it would probably be a very similar scenario with in my postdoc with the beta-2 adrenergic receptors, and yes, I don't know how I've learned this, but it's one of those experiments again. You're looking for something else, and something else actually happens, and that's kind of had that kind of sparked the rest of what I did in my postdoc. So when I was starting my postdoc, Mark's very open. He kind of lets you kind of, what do you want to do, and you can kind of go and explore. And so it was just trying a few different things and stumbling across looking at beta-2 adrenergic receptor endosomes and trying to look at nanodomains. I came across of how this other protein might regulate the recycling of this receptor. So it was, again, it's just one of those moments you're looking for something else, but other things kind of creep up. So I think, yeah.
It keeps us busy for sure. And I think that's one of the difficulties when you're a scientist, is really you have a limited resources, whether it's students in the group, and you have limited time, and then you have to figure out, and sometimes I think the choice is so difficult to say, no, we're going in this direction. And
Yes.
That's when you say, well, actually this we could collaborate with somebody and explore that, but we're going in this direction.
Absolutely. And you also feel you don't want to get sidetracked with what you might be worried about might be phenomenology as well. That's going to end up like a road to nowhere. Yeah, so it's that kind of balance. And I think actually having maybe kind of like as kind of more than one thing on the go has kind of always been a good thing. Have your backup project. It might come your actual primary project, but yes.
Yeah, definitely. All right, last but not least, if you have job openings in your team, where can people find you?
For sure, those will be advertised through the college, Imperial College website. But if any openings, I would absolutely be tweeting it out, and through LinkedIn as well, for sure. But I'm also open if anybody wants to email. Please do so. I did as a postdoc, so I didn't wait. I just went and started emailing people. I thought, oh, this is really cool, and hope that they responded back, which they did.
Yes, I think it's a big part of that. It's just going out there and asking questions, talking to people, and I think it goes back to two points that we covered, is really finding your village and finding your mentors, and you can't do that if you don't reach out to people. But at the end, on the other end, is also finding your next move, and by talking to other people, you can figure out where you want to go.
Completely, yes. And I understand sometimes that kind of reaching out can be quite intimidating, kind of a bit unsure, but people have hesitancy to do so. But in my experience is that people are generally very receptive, very welcoming, and they're not going to have a reaction that you think in your head that maybe that people are. That's just the conversation in your head that's going on. But people want to help. It's all, I think in this field, it's definitely all about pay it forward. So you've had people who helped you, and you always want to help the trainees that are coming through. So it's
exactly. Yeah. I think you made a great point, and I think it's a nice way to close our conversation for today. But yes, I find that the GPCR field it's a cohesive group. You can send an email to anyone. They may or may not respond, but that's typically not personal. They're just busy. And sometimes with all these inboxes and now being able to meet in person with COVID, I think the inboxes have been just exploding. Mm-hmm. But just send the email. Worst that can happen, you won't hear back. That's the worst that can happen.
Absolutely.
Or sometimes you will hear back 2 months later saying, I missed your email. How can I help?
Yeah. So it's okay to prod people as well if you haven't heard back. Yeah.
Exactly. No one will come knocking down your door saying, why did you send 2 emails or 3 emails in the past 3 months?
Exactly.
All right, super. Thank you, Aylin, for your time. I really enjoyed our chat, and I hope to see you soon.
Thank you so much, Yamina.
Thanks. Bye-bye.
Bye.
Thank you for joining us and listening to this podcast episode. We'd like to thank our guest, as well as our team members, Attila Forrest and Ines Pinero. Please make sure that you subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, please leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials. Another great way to support us is to share your favorite Dr. GPCR program with your network and colleagues. Don't forget to check out and register for the Dr. GPCR ecosystem at drgpcr.com/ecosystem. Email us with any questions or suggestions at hello@drgpcr.com. Until next time, stay safe.